A very cheerful morning to everyone. I, Dr. Ankita Tiwari, would like to take the opportunity to welcome our esteemed speaker for today, Professor Chaitanya. I would also like to extend a very warm welcome to all the delegates who are connected to YouTube Live. It is an absolute delight to welcome you all to this day 4 of this enriching faculty development program. Each day has been a stepping stone towards broadening our knowledge, and today is no exception. As we continue this journey of learning, let's embrace the opportunity to exchange ideas, explore new perspectives, and strengthen our professional growth. We are grateful for your dedication and active participation, which makes this program truly impactful. So let's gear up for another day of inspiring session and fruitful discussions. Dear participants, we value your engagement and encouragement to share thoughts or questions with us. Please feel free to drop your queries in the YouTube comment box and they will be addressed at the end of the session. So let's keep this interaction lively and insightful. It is a great privilege to have with us today an eminent speaker, Professor Chaitanya, who is a distinguished academician and researcher with over 15 years of experience in research, academia, and herbal sector. He earned his MPharm in phytopharmacy and phytomedicine in 2008 with an outstanding 82%, earning a gold medal, and he completed his PhD in the year 2017 from JSS Academy of Higher Education and Research, Mysuru, India. During his PhD, Dr. Chaitanya served as research associate at DST SERB, where he developed cost-effective flash chromatographic techniques for extracting polyunsaturated fatty acids, resulting in significant publications and 2 patents. Dr. Chaitanya has received prestigious awards, including the Indian National Science Academy Visiting Fellow Award and the DBT Wellcome Trust Award, Indo-Africa Visiting Fellow Award in the year 2019. His international collaborations include research on anti-cancer properties of Rosmarinus officinalis at the University of Ghana and studies on traditional plants from India and Africa with anti-TB potential at the University of Toroya, South Africa. A pioneer in herbal drug discovery, Dr. Chaitanya specializes in phytochemical research using advanced techniques like HPTLC, HPLC-MS, GCMS, and flash chromatography. His work bridges traditional botanical knowledge and modern science to develop sustainable medicinal solutions. In addition to his research, Dr. Chaitanya serves on editorial boards and acts as an independent consultant, advocating for environmentally conscious practices in natural product chemistry. His contributions aim to integrate ecological mindfulness with advancements in herbal medicine. Currently, he is working as a professor in School of Pharmaceutical Sciences, Lovely Professional University, Punjab. So with this, I would like to invite Sir for his talk. Over to you, sir. Thank you.
Thank you, Dr. Ankita, for the nice introduction. Okay, so thank you, Dr. Rakesh, for giving me this opportunity and support and screen. Okay, so I hope the rights are with me.
Hello.
Whether the screen is visible right now?
Yes, Professor Chaitanya. Good afternoon.
Good afternoon, Dr. Rakesh. Can I proceed with this?
No, screen is not visible.
Not visible.
Please share your screen.
Okay, is it visible?
Yet not.
No, sir, not yet.
I am sharing.
Yeah, it's
Yes,
visible.
yes, now visible.
Now visible. I shared. And can I proceed with this?
Yes.
Yeah. So good afternoon, all. Okay, so I'm here in front of you to deliver my natural product drug discovery journey. Okay, with some examples, live examples. Okay, so this focuses on the recent insights in your natural product drug discovery and development. As we know, even we have hit with many diseases recently, which are a problem with our immunity and problem with our dietary and environmental issues. A recent issue, I can say you two examples. One is COVID 19, and another one is our dengue fever. Okay, these two things, when they have hit as a pandemic and endemic, we don't have any drug in the market. So at that time, we realized even during the COVID 19 times, we don't have any immunomodulator in the market that should be available at an economical rate to all the individuals. But at that time, there is one plant which you can call as Godichi, which you can call as Tinospora cordycefolia. And even when the dengue has hitted, okay, we don't have any other molecule apart from your Carica papaya. Okay, today also, like some 3 years before, Avantis has launched this product, okay, as a standardized Carica papaya leaf extract. And today we have depend till today to increase our platelets. We have depend upon the nature. This is only some of the recent examples, but these examples have started long back in 14th or 13th century. Taking an example of morphine in our 18th century, and salicin in our 18th century, tubocurarin in our 19th century. Until today, for cancer, we are saying only one molecule, that's all. Okay, so this taxol is present and we are synthesizing it, but the lead molecule always got from the plant called Taxus brevifolia. Okay, so with this brief introduction, I'm going further to start. Today, an innovative talk on recent insights in natural product drug discovery and development. Okay, so if you see this natural product and their challenges, okay, always there is a need for a new molecule, which you can call as the drug lead or a drug. The journey starts not within a year, from it will take around some 14 to
Chaitanya
15
sir,
months.
excuse me.
Yes, sir.
Please make it slide show, na? Your
Is
presentation. Yet,
is it moving?
yes, it is moving, but make it in slideshow move.
Okay, slideshow mode. Okay.
Hmm.
So you may click on that slideshow option at the top
Yeah, yeah.
menu bar and
Is it
from the
okay?
current slide.
Please
Yeah, is it okay?
double
I
click.
have kept the slideshow.
Okay, slideshow.
Hope is it okay?
Visible. Option at the top bar and from the
It
current
is
slide.
visible. Okay. So, if you see the importance of the natural products and their challenges, there are many challenges. Okay, and there are many problems. But for all the problems of the drug discovery, we are having one solution, that is Mother Nature. Okay, always the drug discovery, it will start only by means of isolation, characterization, optimization. Okay, and we got certain type of resistant diseases like cancer and tuberculosis. Till today, we having no drug to address this resistance. In order to address the resistance, we are having only one solution. The solution is Mother Nature. Let it be a plant, or let it be a microorganism, or let it be a algae, or let it be fungus. But always this new molecules or the new chemical entities, okay, the first structure is from our natural library only. Okay, so these are all some of the examples where the journey of the drug discovery started from 18th century and it is going on till today. And by 2030, the natural products in the form of dietary supplements or the functional chemicals, it is carrying a lot and huge CAGR. So there is a huge demand for the natural products, but this huge demand is not that much simple to address these issues. Okay, there are a lot of challenges, there are a lot of complications, there are a lot of complexities. Always the synthetic drug discovery is too easy, but the natural product drug discovery is high demand, but it is little complex. Okay, in order to address this complexity, definitely we need to have a recent developments in the sense of chromatography and the flash techniques. Okay, so if you see the whatever it is, the natural wherever we are saying, the ultimate end in the natural product drug discovery is the phytochemical. Okay, so the phytochemicals, if you see, we got secondary metabolites, we got primary metabolites. Okay, till today, many wonderful drugs have given from an moiety or the group which you can call as alkaloids. Okay, even these alkaloids are wonderful drugs, but the plant synthesizes this molecule, okay, by means of secondary metabolic pathways or by means of primary metabolic pathways. There are wonderful pathways, even it is there in our bform syllabus, stating that acetate malonate pathway, acetate malonate pathway. These are our wonderful pathways, and even recently, if you see the insight, in order to address this complex libraries, we have come up with a very innovative technique which you can call as radio tracing techniques, okay, which is also very useful. Okay, the drug discovery process, even if you see 90% of the drug discovery process, still we are enjoying the status of a drug in our monograph. It is a product of ethnomedicinal drug discovery, or you can call it as traditional knowledge. There are few countries where they got an extensive traditional knowledge. In that, one of the country I can say proudly is our India. Okay, so totally it will take around some nearly 14 to 15 years. Okay, but mainly the problem is most of these drug discoveries are stopping at the preclinical level in the academia only. We are not focusing them to commercialize it or to carry forward once our project is completed. But many of these have got, maybe I can say, these drug discoveries are stopped due to lack of funding or due to lack of facilities, okay, to address this complex nature. Okay, however, out of thousand of the molecules every year we are getting hardly around some one or two molecules as the drug. This is a highly challenge, but the disease resistance is going very faster, but the drug discovery is going very slow. This is one of the important thing where we need to address. Okay, so there are several technologies in the drug research. Okay, but nowadays, if you go to the industry level, the drug discovery always it will start from the extraction, but no one will do the soxalate or no one will do the microwave. Okay, today the wonderful equipment where the industries are using is these are all the wonderful equipments at a pilot scale, large scale, and a small scale. Okay, one of such a thing, if you see, nearly 70% of the extractors in a very good MNC herbal companies have been replaced by supercritical fluid extractor. Okay, so this is one of the important thing, and we got one of the thing by Buki or the Buchi, which you can call as an universal extractor. Okay, this universal extractor can also act as a fractionator. Okay, at per 30 minutes, you can get around some 200 to 300 different polar fractions. Okay, so this is one of the important thing. And even the third diagram you are seeing, that is a commercial fractionator. Okay, and today I said you like one of the important thing in the drug discovery is bioassay guided fractionation. But doing the assay guided fractionation in a separating column or the column chromatography, it will take lot of solvent and it will take lot of time. So we need a flash technique where we can get around some 100 to 150 of our samples per 30 minutes. Okay, so the third example is that these are all some of the latest insights in our herbal drug extraction. Okay, latest technologies, even like, and today we are in an era, okay, not saying about your only liquid, but we are saying and conjugated techniques or the hyphenated techniques, where we need a conjugated techniques with the chromatography and the mass spectroscopy. And today we are speaking not one LCMS, but we are speaking two MSMS. Okay, in order to address these complex molecules, okay, we need definitely sophisticated instruments. These are all some of the sophisticated instruments. And recently, if you see, even HPTLC, I think like 3 to 4 years before, we came up with even a conjugated system called as HPTLC-MS, which is a very wonderful system. But only the challenge already I'm saying, these are all not simple. We need a huge amount of money. You need a huge amount of funding. So this is only the challenge. Okay, so this is a GCMS. Okay, there are several type of spectral techniques, and even if you want to finalize the structure, how many spectroscopies we having also, we need to have NMR to come discover. So these are all some of the examples of the drug leads to the drug. Even you can take the best example. Always I will say every seminar that is an anti-cancer drug. You can take a Taxol, etoposide, camptothecin, and even malaria. Okay, so if you see like malaria, India is okay, it is lesser prevalent. But if you take the countries like Africa, it is a very highly prevalent, and Plasmodium falciparum is a highly challenging microbe, and due to its resistance. Okay, the story of earthy ether and earthy sulfate came into the picture of the microbial resistance, but only the Africans having no knowledge to create or to isolate this drug. They have depended upon two countries. One is our India and another is our China. Okay, so this is the thing. Okay, these are all some of the examples, and even whatever the disease you say, I will say like this is the lead where you got it. Okay, and we have to be pride enough that these leads are coming only from our natural product through the drug discovery and development. Okay, so as I do several times, okay, so even if you say the nature is complex. In order to break that complexity, we need a technique and we need a knowledge of polarity. Okay, so one molecule will be one polar, one solvent where we can call as polar, non-polar, and semi-polar. These are all some of the examples. And whenever you want at the drugs like glycosides, alkalides, and fatty acids, the best solvent to go for it is always the chloroform and dichloromethane. So these are all some of the examples which will be useful when you started your bioassay guided fractionation. Okay, next. These are all some of the targets. See, today we are at a level. If you say like I will do the extract, I will do the fractionate, I will do some microbial activity, or I will do some anti-inflammatory activity, means no one will accept even our papers. Okay, no one will accept even our research. They need definitely which molecule is responsible. Okay, so definitely we need a molecule and we need a molecular target. So if you see, these are all some of the important targets. Being I'm a cancer researcher, I'm showing only the important cancer targets. There are nearly 30 to 40 important targets are there. Out of that, recently EGFR is gaining an very good importance. Okay, and even VEGF is also gaining a very good importance. Even though there are topoisomerases are there, caspases are there, PARP inhibitors are there. And even we are not speaking about that. And today at 21st century, we need to say not only these targets, but we need to say epigenetic targets. We are at a level to say and analyze how our drug will act on genesis and epigenetic targets. Okay, the scope, there is a huge scope.
Sir, sorry to
Yeah,
interrupt
no
you. So
problem.
your slides are not moving.
Moving, but in my it is moving.
Now it has moved, sir.
Okay, so if I go to the slideshow mode, the slides are
Yes,
not
sir.
moving.
Uh, if you could please press Shift and F5, that will automatically take you
And
to
F5.
slideshow.
Okay, I will try that.
Thank you.
But my voice is clear.
Your voice is absolutely clear, sir.
Okay, thank you so much. Okay, fine. So if you see the scope, there is a huge scope. Okay, and I said you like it is going to share a CAGFR of 25 to 30 in the few years. Okay, in that where we have to address many resistant diseases like antimicrobials, anti-cancer.
But where we need to get these molecules will get only by means of natural product drug discovery, where we need to have an extensive knowledge on pharmacognostical technologies. Okay, pharmacognostical technologies knowledge is a very essential, and it is a good research gap that this knowledge is going to what will say, like, it is lowering in the coming years, okay, due to lack of awareness and whatever it is. But the drug discovery always start only due to our pharmacognostical knowledge, not any other knowledge. Okay, so drug discovery, I said, one of the important cases, if you see recently, by 2030, and it is going to reach around $32 billion. Okay, it is called as fatty acids. Okay, and we know that nowadays the food whatever we are taking is not sufficient. We need to have antioxidants, we need to have functional chemicals. Okay, out of that, if you type a simple thing in Amazon, okay, all these dietary supplements, they need to have only one thing, what you can call as polyunsaturated fatty acids, which you are calling it as PUFAs. Okay, it is a high demand, but these PUFAs are highly rich in some of the natural products. You can take the example of spirulina, which is released into the market as a single cell protein. But if you see this spirulina today, it got a wonderful PUFA, what you can call as gamma linolenic acid. Okay, when I am doing the research on the spirulina, we have found like the spirulina contains around 18% of gamma linolenic acid. 1 ml of gamma linolenic acid today, the cost by your Sigma Aldrich is 35,000, and even the stability issues are there. Okay, so, but there are several examples. Okay, not only your spirulina, there are certain type of mushrooms, even though these PUFAs are very high. And one of the wonderful fruit, I can say you, as a good source of your fatty acid, is butter fruit, which you can call as avocado. In India, it is costly, which each fruit goes around 100 to 70 rupees. But in Africa and other places, it is very cheaper, and it costs around some only 5 rupees if you convert into your Indian currency. Okay, in Africa, if you go, every house they will have one avocado tree like your mango tree. They know that it got its own nutraceutical value. Okay, so you can take some of the examples of my live examples from my team. Okay, even you can see it is a plant which you can call as Rubus stedunary. Okay, and normally you can call it as a cherry plant also. Okay, so this is a small research which we have carried out in Africa. Okay, with my collaborators in Ethiopia, and we have found these fruits are very good source of a certain type of fatty acids. In fact, we are the first team to say like these fruits are rich source of butyl isobutyl phthalate, which is also a fatty acid. Okay, this is one of the example. Okay, and even if you see, this is the process usually nowadays. In order to speed up our drug discovery journey, definitely we need to have a flash knowledge. Okay, so no one will use the traditional column chromatography, which will take years, months to get one molecule. But if you know a robotic flash, it is very easy to discover our new molecules. In that journey, at a preclinical level, I can say this is one of the important example. Okay, the plant is called as rubus, okay, which is identified and collected in Ethiopia. And we have found it is a very good source of PUFAs. But we have to know how to isolate these PUFAs. PUFAs are highly challenging compounds. Okay, so
So, sorry, could
yes,
you please move it from the left side of your screen?
okay, left side.
Still the slides are not moving, sir.
Okay, is it now?
Not yet, sir.
But there is some problem. I don't know. It is moving in my screen.
And now it's moving, sir.
Yeah, because the problem is so this.
It's okay, sir.
Okay, can I proceed?
Yes, sir. Sure.
Okay, so these are all some of the important leads which we have discovered from the plant called rubus. Okay, we have found these rubus fruits are highly rich of nearly 25 very good fatty acids. Okay, out of that, even we have found there is a lead of gamma linolenic acid. Okay, so this is one more journey. Okay, so this is recently published also in Pharmacognosy magazine. Okay, and if you see one more drug, I will say like it is which is in a huge demand, but there is no research data. Okay, so all researchers can focus on this herb. It is rosemary. Okay, so the research is saying like if you sniff, there is no need always the drug means it is not an injection or tablet. Drug means even by eating you can heal, by smelling you can heal. Okay, that's why even for your cancer therapeutics, okay, the recent approach by WHO is saying like don't overburden the patient already who is overburdened. Okay, so definitely we are looking for certain type of aromatherapy healing agents through nasal delivery. Okay, in that one of the important we found rosemary. Okay, so rosemary for your cancer therapeutic, it also got it as immunomodulatory activity and also it also got memory enhancing activity. Okay, so the best example you can take it as an example. No need of going a big lab. You can take a rosemary herb, daily sniff it means smell it nearly some 5 minutes. You can see at least within 6 months you will see like there will be around 13 to 15% of your memory enhancement. Okay, this is scientifically proven. Okay, so this is one of the example rosemary we have taken and we have developed an asava from the rosemary, which is the first time in its nature, and we have subjected it to certain type of in silico screening, and we have found like this rosemary is an active and it's showing a very good IC50 values, okay, on nearly 17 cell lines including your breast cancer, cervical cancer. And people say like it is due to your rosmarinic acid, but it is not due to rosmarinic acid. We have found like there are many other wonderful compounds which is showing more potent than the rosmarinic acid. Okay, such an example is rosemary quinone, rosemanol, rosmadiol. Okay, all these things are we have found only in the Rosmarinus officinalis. But till today, very less data is available on the molecular mechanics of your rosemary quinone and rosemanol. Okay, restricted only to the research that is available over the web on rosmarinic acid. But we have found this rosmarinic acid is okay, fine, but there are several molecules, okay, which are very potent than your rosmarinic acid from the same herb. Okay, so this is some of the examples of the research publications from the herb which we have started in our laboratory, the journey. Okay, next you can take the example of genicity. This is a wonderful molecule which I find. Okay, and if you know, like the North India, we love like soya chaps and soya. Okay, daily we'll say soya. Soya is a very rich of genistetin. Today, genistetin is sharing around 13.5% of CAGR. And today, genistetin is a very wonderful biofunctional supplement that is showing and complete a solution for all your inflammatory diseases. Okay, but there are several sources, okay, that having genistetin. Okay, even in my live examples, we have found like there are certain type of weeds which grows unnecessarily on several areas. Okay, and it is a very cheaper source, no need of cultivation also. Okay, we have found even genistetin is present in many weeds. Okay, so this is the thing. And this is an investigation which I have found like the genistetin, okay, by simple LCMS, by means of its chemotaxonomy, comparison with the library, we have found like there is a weed which you can call as Scotch broom. Okay, but there is no data that is available on the Scotch broom. Okay, there is no research. Okay, we are the first team to isolate or identify the genistetin from Cystis scoparis. Okay, but we are unable to publish in a very good high impact journal due to various factors. Okay, so this is the thing. And even the story of tricaprion. Okay, so if you take this tricaprion, the tricaprion is a very wonderful plant. And those people who are from Kerala, if you say like there is one plant called Lakshmi Taru, which you can call as Semaruba glauca. Okay, when we are doing the Semaruba glauca, and today if you type in YouTube and you are seeing like anti-cancer drug discovery, all this anti-cancer drug discovery, you can say like there are many hundred of videos that are available on only on this plant, which you can call as Lakshmi Taru, okay, which you can call as Semaruba glauca. Okay, we are the first team to isolate an PUFA again, which is a tricaprion, and we have proposed an mechanical studies, not only restricted to the in vitro studies, and we have proposed even a molecular mechanism studies first time. Okay, we have found like it is a very good class 1 histone deacetylase, which is a very good epigenetic target also. Okay, so this is one of the example which we have seen. Tricaprion. Many plants they contain tricaprion, but this molecule we have neglected this molecule. Okay, so we don't have any big data or research that is available, but it is a very potent plant, a potent molecule, and it is a very potent plant. In Kerala, if you say like there are several traditional healers, they will take these aerial parts and they will boil the decoction and they will give to the cancer patient. Okay, so this is the story. There is a wonderful story. Methyl gamma linolenate, or you can call it as gamma linolenic acid. Okay, it is a single molecule that keeps a full stop to all your oxidative stress. And today, even if you see those who are having diabetes, even in the diabetic prescription of your multifunctional supplement, they are that is your methyl gamma linolenate. Okay, so but we got many other sources where we will get the methyl gamma linolenate. Such type wonderful journey, you can say like olden days, even we can call as spirulina platensis. Change, we have to say it as only Orthospira platensis. Okay, it is an first flash chromatography I have done. Okay, I have tried only on this plant or algae, and we are able to isolate and identify nearly 23 potent molecules from that. Okay, due to lack of funding, we have focused only two important PUFAs from that. One of the important PUFA is gamma linolenate, and its bioactive form we have semi-synthesized, and it is called as methyl gamma linolenate. These are all some of the examples how the drug discovery will start. Okay, next is the chlorogenic acid. Even if you see the chlorogenic acid, today this chlorogenic acid is a wonderful molecule. And today we are saying like the coffee. Okay, coffee is a very good, even nowadays, even I'm seeing one article 2 days before, having coffee will protect your fatty liver. Okay, and it is not due to your caffeic acid or other phenolic acid. It is due to a neglected molecule which we have not spoken and given focus from the long back, which is called as chlorogenic acid. Today, chlorogenic acid is a wonderful CAGR is 3.89%, means it is a very use. It is a very good dietary functional supplement. And there are several molecules, even several dietary rhizomes, which we don't know. They contains chlorogenic acid. And even our coffee daily we are having, it is a rich of chlorogenic acid. Okay, so this is one of the important thing. And we have done a plant on the Lycia spinosa. Recently it has published even the Lycia spinosa. Okay, so in the journal called as Natural Product Journal, I think so. It is from Time Connect or Willy. Okay, so this is the one of the wonderful journey which we have given. And if you see like this is the story of Lycia spinosa. Okay, we have done many formulations from that also. Okay, this is if you go to the region called Vizag or the Visakhapatnam, the tribal people will use this rhizomes. They will clean it and they will wash it, they will boil it, and they will have it. Okay, so for many disorders. Okay, one of the such ethnomedicinal evidence when we are interacting with the tribal people, they said like if you have and if you eat it, our colons will be cleansed. They have said like our digestive disorders will be healed, and in that journey we have brought that and we have screen and bioassay guided fractionation, and we have found like it is not only healing a single cancer like your intestinal or colon cancer, it is healing and showing a very potent IC50 lesser than around 45 to 35 microgram per ml. Okay, on nearly 13 cell lines. Okay, but if you see the research is very limited, we are having a very scope to carry out further research to propose or convert these drug leads into the drug. Okay, so next, quercetin. Today we know like, today the era of 21st century in the form of functional supplements, and we are saying like senolytics. Okay, in a normal terminology, we are calling it as, I can say the anti-aging drugs. In a scientific terminology, we are calling it as senolytic. Everyone will say like resveratrol, but resveratrol is a very highly costly and we don't have any clinical evidence that it is a senolytic. But we are forgetting a simple molecule which is cheaper, that is quercetin. Okay, this quercetin is present not only in fruits, there are many weeds which contain quercetin. Why can't we isolate the quercetin and why can't we commercialize it from the cheaper source? Okay, so these are all some of the examples. Okay, so you can take the Erigeron bonarensis. It is a very one of the example, but there is one more plant which grows called as Erigeron carvancianus. Okay, even that also contain a high amount of quercetin. You take this Solanum maritonum. okay, which will grow on many areas, nearly hill stations, I can say, as a weed. If you go there, it will cause itching also. It is little bit toxic, like your parthenium. Okay, it's a weed discovery. We have found like there are many wonderful anti-cancer drugs in that, but still we didn't carry forward. Only our research has restricted only to our LCMS fingerprinting and chemotaxonomic fingerprinting. This is one of the wonderful thing I am saying, and this flower is called as daisy flower. Okay, you have seen it in many of the houses. They will cultivate it, or they will use for your ornamental purpose. And this contains a very wonderful antifungal compound which you can call as calenduloside, a very wonderful anti cancer compound which you can call as leocarposide. Naringenin is there, aspiritin is there, and these are all wonderful molecules which is present in the chemotaxonomical libraries of the plant that belongs to Asteraceae. Okay, so these are all wonderful journeys, and you can take the example of ruscogenin again. This ruscogenin is shown in our preliminary study on one of the breast cancer cell lines at 15 microgram per ml, but we are unable to carry forward this research due to the lack of funding. Okay, so these are all some of the examples. Even saponins. Today, saponins are very used. Whenever you want an pull stop to your inflammation, okay, one of the molecule I can say is the saponins. But isolation of saponins is not that much easy. It need lot of practice. It need lot of talent. It need lot of skill. It need lot of complex equipment. Okay, these are all some of the docking which you can say. Without docking, even we can't say, but by docking we can say our conformance. There are many failures. Even docking shows a very good result, but when we are doing an molecular mechanism studies, our results are fail. Okay, so to conclude, definitely we need to have a good knowledge of our pharmacognosy or our herbal drug technology or our natural product to address many diseases that are challenging even today in the 21st century. So this challenge can be addressed only with the pharmacognostical knowledge, okay, and from our natural product libraries. And this drug discovery journey, which has started in the 14th century, not even stopped till today. Okay, till today we are searching many mushrooms, we are searching many algae, we are searching many animals, we are searching many insects, and we are getting new molecules. Okay, so this is the thing. Okay, so in every walk with nature, one receives far more than he seeks. So nature is having many hidden secrets. Being a phytochemist, I love to explore the hidden secrets in the form of phytochemicals. Okay, with this, I'm pausing or I'm winding up my session. Okay, now the session is open for the discussion. I am thanking the organizers to give me this opportunity.
Hello, Dr. Chaitanya.
Yes, sir. Yes, sir.
Yes, sir. Good afternoon.
Yeah. Good afternoon, sir.
So thank you very much for giving such a very nice lecture. You have started this lecture from primary plant metabolite, then secondary plant metabolite, then goes to the biosynthetic pathways. Very beautifully explained. And after that, you give some case studies with your foreigner collaborators, and you explain each and all aspects of your studies, and you suggested the uh, about the various fact about the which we can include in our research work also. So thank you so much for giving such a nice presentation and sharing your expertise with us.
Thank you, Dr. Rakesh, for these beautiful words.
Ankita.
Thank you so much, sir. As the word Chaitanya reminds us that continuous development begins with conscious awareness and purposeful action. So thank you so much for your exhaustive deliberation, and I hope that all the researchers working on herbal products might have been benefited by your lecture. Thank you so much,
Thank you,
sir.
Dr. Ankita.
I cannot find any question. Your talk was very clear to all the participants.
Okay.
Yes, it was very commendable and
Very explanatory, sir.
Okay.
So thank you so much for
So
sparing your
thank you, Dr. Ankita, and thank you, Dr. Rakesh, and thank you to the team of Sharda University for giving me this opportunity. Thank you all.
pleasure is ours.
Okay.
Bye. Take care.
So, as we wrap up day 4 of this enriching everyday, we reflect on the wealth of knowledge shared and the engaging discussion that was unfolded. Today has been another step forward in our collective journey of learning and growth. So, let's continue this momentum as we prepare for the final day tomorrow. Until then, all of you, please take care, stay inspired, and have a very wonderful evening. With this, I would like to say goodbye.